Hello, my name is Nate. If you've clicked on this video, that means you've clicked on the shortcut version of the pyruvate dehydrogenase complex in fermentation. All of this information will be included in my longer metabolism video as it gets finished, just in case you're wanting to watch all of these in a row in one concise video. You can also check out my website for other free resources, tips, tutoring access, a whole bunch of other things like that. All of that will be linked in the bio, as well as some notes to support all of the information that's on these slides, just to make your life a little bit easier for note taking in case you want to use any of my graphics, anything like that. So with where we just left off, we have just turned the 6-carbon sugar glucose into 2 separate 3-carbon pyruvates. We've basically split glucose in half to produce 2 different pyruvates in a process called glycolysis. And in doing so, that will have produced a net of 2 ATP and 2 NADH in addition to the pyruvate. This is where we left off. So if you're looking at what's next, it's going to be an enzymatic complex called the pyruvate dehydrogenase complex. Basically, we're going to keep turning this molecule, this carbon chain, into different molecules in order to keep exacerbating all of this energy out of it. We're going to keep producing ATP and the electron carriers in order to produce the most ATP possible. The electron carriers eventually down the road will be sent through the electron transport chain, as you can see towards the bottom of this pathway, to produce ATP. Before we dive into the pyruvate dehydrogenase complex, what exactly we're going to be doing with this pyruvate, we need to talk about the cell and the cell structures. Remember that we are looking at a eukaryotic cell, and as we go through metabolism, everything that we're talking about is eukaryotic metabolism, unless I specify otherwise. The first portion of the cell that I want to point out is the cytosol. The cytosol is the fluid in the open area around the inside of the cell. It's basically, if you're sitting in a room, it's the air around you in the room. That is the cytosol. It is interchangeable with the word cytoplasm. They're both essentially the same thing. One is a fluid, the other one is the area the fluid sits in. The cytosol is considered to be anaerobic, meaning that oxygen is not present or required in the cytosol. So any processes, such as glycolysis, that occur in the cytosol, we consider them to be anaerobic and they do not require oxygen. We're going to be able to run those processes whenever we don't have oxygen. Additionally, the cytosol is a relatively reducing environment. Molecules that are in the cytosol, they are going to want to be reduced because of all of these reducing agents that are going to be floating around the cytosol. Glycolysis, just as a reminder, will be occurring in the cytosol and therefore is anaerobic. The next structure that we need to talk about is the mitochondrial matrix. The mitochondrial matrix is the open area on the inside of the mitochondria. Notice in our mitochondria, we have this nice membrane that is separating the inside of the mitochondria, aka the matrix, from the cytoplasm which surrounds it. The mitochondrial matrix is considered an aerobic environment because oxygen is going to be both present and required for all of the processes that occur within the mitochondria. Anything that occurs in the matrix is going to be considered aerobic, meaning oxygen in some way is either required or has to be present. The pyruvate dehydrogenase complex, conversely to glycolysis, will occur inside of the mitochondrial matrix. Because of that pyruvate that we just produced in glycolysis in the cytosol, and additionally some of that NADH that's going to have to get to the electron transport chain, we'll talk about that later, they are going to have to cross the mitochondrial membranes. Crossing a membranes, unless you're just a super small nonpolar molecule like most of our gases, oxygen, carbon dioxide, etc., which pyruvate is not, is going to require some form of energy. Luckily, for pyruvate, you don't need to know the exact way that pyruvate moves into the matrix. You just need to know that moving pyruvate across a membrane is one of the ways that we can lose a little bit of our ATP yield. Because eukaryotes have these membrane-bound organelles, we're going to be losing a little bit of that energy yield in order to get things into the right place. This is an issue that prokaryotes do not have because they do not have any membraned organelles. All of their organelles are just sitting out in the cytosol, They don't have to worry about getting pyruvate to cross a membrane. Again, we will cover the differences between prokaryotes and eukaryotes at the end of aerobic respiration. So now that we have moved that pyruvate into the mitochondrial matrix, we can now send that pyruvate through what we call the pyruvate dehydrogenase complex, the PDC for short. The PDC occurs within the mitochondrial matrix of most of our human cells. There's one important exception to that for the purposes of the MCAT, and that is our red blood cells, RBCs for short. Red blood cells, pretty much the only thing that they do is they carry oxygen. So they are not going to have any mitochondria. Therefore, all of this aerobic respiration, starting with the pyruvate dehydrogenase complex, they will not have. All of our other cells, conversely, are going to be able to conduct the pyruvate dehydrogenase complex and the rest of aerobic respiration. As I mentioned on the last slide, because the inside of the mitochondria is aerobic and requires oxygen, the pyruvate dehydrogenase complex is also going to be considered aerobic. The net reaction, or the goal of the pyruvate dehydrogenase complex, is we are taking one pyruvate, and that one pyruvate will undergo a redox reaction to produce one NADH, one acetyl-CoA, and one carbon dioxide. In other words, a 3-carbon pyruvate is going to lose a carbon in the form of carbon dioxide. And this is going to help us produce acetyl-CoA, a 2-carbon molecule, and give us the energy to form NADH. Pyruvate will be oxidized, NAD will be reduced into NADH. As another reminder, it's important to remember that this reaction is going to be happening twice per glucose. Since every single time we run one glucose through glycolysis, it's going to be producing 2 pyruvate. So you're always going to want to make sure you're keeping track of the numbers on a per glucose basis as well as a per pyruvate basis. So per glucose, 2 pyruvate will produce 2 NADH, 2 acetyl-CoA, and 2 carbon dioxide. When does the pyruvate dehydrogenase complex occur? It's going to be promoted in these fed postprandial conditions when our glucose concentration is going to be very high, and this is going to lead to a high concentration of pyruvate coming through glycolysis. And additionally, in order to actually start the pyruvate dehydrogenase complex, the mitochondria has to have oxygen. So not only is the pyruvate dehydrogenase complex going to occur when we are in the postprandial state, it also needs oxygen to be present, and we also can't be super desperate for ATP. When we are running the PDC, we are choosing to prioritize efficiency over speed. This is going to be really important whenever we talk about fermentation after this. The main enzyme that you need to know is simply pyruvate dehydrogenase, thus the name of the complex. It's the main enzyme in this big complex with 3 other peptide subunits. One important note that you need to know about the PDC and pyruvate dehydrogenase is that it requires what's called TPP. This is a derivative of vitamin B that is considered a prosthetic group. This is basically just a type of cofactor, some sort of separate molecule that is going to be required for the proper functioning of the pyruvate dehydrogenase complex. Basically, if TPP is not present, the PDC is going to have issues running properly. The hormone that will end up promoting the PDC, of course, is going to be insulin. Insulin promotes glycolysis. This is going to increase the pyruvate concentration, and therefore we now have way more of the reactant of this reaction. So, of course, it's promoted by insulin indirectly. It is also going to be promoted by low energy, low ATP concentrations, as well as high glucose and high pyruvate concentrations. That should all be pretty self-explanatory. We want to be doing this when we need ATP and we have a lot of glucose and pyruvate. It is going to be, of course, inhibited by glucagon. This is also going to be indirectly. There's no direct action for glucagon to specifically inhibit the PDC. But again, for the MCAT, you just need to know that glucagon inhibits the PDC. It is also going to be inhibited by high energy levels. If we have a lot of ATP, we don't need to be producing more through the rest of aerobic respiration, including the PDC. And additionally, High amounts of acetyl-CoA or citrate will also feedback inhibit the PDC. If we have a lot of product, we don't need to keep running the reaction further on. Citrate is going to be the first molecule in the next step after the PDC. It's called the Krebs cycle. So if we have a lot of citrate, we don't need to be making any more. A quick note to start introducing this idea of why the speed and oxygen presence is important for these processes. We've mentioned that glycolysis is anaerobic, meaning that it does not use oxygen, and it is also super fast relative to most of our other metabolic processes. The pyruvate dehydrogenase complex is aerobic, so it's going to require oxygen, and it is only going to be used when we don't need ATP quickly. It's only going to be used when we can prioritize efficiency. The pyruvate dehydrogenase complex, as well as the rest of aerobic respiration, is going to be relatively slow. It's very efficient, meaning we can produce a lot of ATP per glucose, but that efficiency takes a long time. So, why is that? We get to find out on the next slide. So on this slide, we're going to be introducing this idea of why the oxygen presence and speed actually is going to be pretty important for our cells. So just as a reminder, aerobic respiration, of course, is going to start with glycolysis, but glycolysis itself is not necessarily aerobic, but it's still included. It's going to run through the pyruvate dehydrogenase complex, which we're talking about now, and then finally through the Krebs cycle and the electron transport chain. It is very efficient to go through this route, meaning it's going to produce a lot of ATP per glucose, but as a whole is going to be relatively slow. If we have low ATP and high glucose levels, meaning we need to produce ATP and we have the glucose to produce it, but we need ATP faster than aerobic respiration can provide, we're going to be using a process called fermentation instead of the pyruvate dehydrogenase complex and the rest of aerobic respiration. So just to compare and contrast these two, we're going to start with the pyruvate dehydrogenase complex on the left, and we're talking through the rest of aerobic respiration as well. We are going to be doing the PDC and the rest of aerobic respiration whenever we are in the fed/postprandial state. We have low amounts of ATP and high amounts of glucose, of course, but our need for ATP is not desperate. That's the word that I'm going to use. We don't need it super quickly, so that's going to allow us to prioritize efficiency. Additionally, we are only going to be able to do aerobic respiration whenever our oxygen levels are normal or high. So for example, we're going to be doing a lot of aerobic respiration when you've eaten a meal and you're just sitting on a couch. We're in the postprandial state. You're not doing any crazy exercise or anything like that. Your body can just relax and do the efficient aerobic respiration. It's going to send pyruvate through the pyruvate dehydrogenase complex. This is going to occur in the mitochondrial matrix, which of course means it's aerobic. And just as a reminder of the speed, it's relatively slow because there's going to be a lot of steps involved throughout the rest of aerobic respiration after the PDC. It is very efficient. It has a high efficiency because we can produce roughly 30 to 32 ATP per glucose. We'll talk about that later when we're done with aerobic respiration, those specific numbers. Fermentation, on the other hand, is going to be promoted by a lot of the same conditions, such as low energy, low ATP, and high glucose, meaning we have plenty of glucose to make ATP, but our need for ATP is desperate. We need it very quickly, faster than aerobic respiration can supply it, or whenever we don't have oxygen. For example, in super intense exercise, there's two issues. We need ATP super quickly to provide for sliding filament theory, basically how our muscles move. Additionally, we need a lot of oxygen in order to keep running aerobic respiration. If we don't have enough oxygen to keep aerobic respiration alive, or our exercise is intense enough that it is too fast, our ATP need is too fast for aerobic respiration, we're going to begin fermentation instead of the pyruvate dehydrogenase complex. Notice that fermentation, instead of being in the matrix, is going to stick in the cytosol. Because of that, it is anaerobic and does not need oxygen to be present. It is also going to be very fast in comparison to aerobic respiration. Even though it is fast and can produce ATP at a high rate, it is producing ATP at a very low efficiency. Even though we can produce more ATP per second, we are producing way less ATP per glucose. This is a very wasteful way of producing ATP. Notice that the efficiency is only 2 ATP per glucose whenever we send pyruvate through fermentation instead of through the rest of aerobic respiration. One thing I want to make a quick note of is the way that we get from lactic acid and ethanol, the products of fermentation, back to glucose is through a process called the Cori cycle. We're not going to talk about that right now. Just go ahead and throw that behind you for a little bit. We'll talk about that after gluconeogenesis. So just to summarize what we've talked about so far, once we have produced pyruvate through glycolysis, our cells have to make a choice. Do they have oxygen, and how desperately do they need ATP? If they've got plenty of oxygen and we're relaxed enough that we don't need to be making ATP at a super high speed, we can run through the rest of aerobic respiration, sending that pyruvate through the PDC. However, if we do not have oxygen, or we need ATP at a super high rate, a super fast speed, we will instead send pyruvate through fermentation, a reaction that will occur in the cytosol, does not require oxygen, meaning it's anaerobic, where we will produce lactic acid. We'll talk about why that's important here on the next slide and why that will end up being faster than the PDC. Now to explain fermentation and this process that we're going to call anaerobic respiration, meaning it doesn't require oxygen. It is going to be taking place in the cytosol, the same location as glycolysis, meaning we don't have to move pyruvate over any membranes. It is going to be anaerobic, meaning it doesn't require any oxygen. And I want to note that combining glycolysis and fermentation is what we consider anaerobic respiration. Aerobic respiration is glycolysis through the electron transport chain and everything in between. Anaerobic respiration, which does not involve oxygen, is going to be glycolysis and fermentation instead. One other thing that the MCAT likes to talk about is the rate of glycolysis versus the rate of fermentation. Notice that because glycolysis is feeding the reactants of fermentation, and through the Cori cycle, fermentation will be sending everything back into glycolysis, the rate of the two is going to be the exact same. They're going to proceed at the same rates because glycolysis feeds into fermentation, which will feed back into glycolysis. The goal or net reaction of anaerobic respiration and fermentation is to take pyruvate and NADH, one of those reduced electron carriers, and we're going to be forming lactic acid and NAD+ in mammals, or ethanol and NAD+ if you're yeast. Unfortunately, I'm not a yeast, I'm a mammal, I have to pay taxes, so we're going to be making lactic acid instead of ethanol. So in this condition where we have to increase the rate of ATP production, we're going to increase the rate of glycolysis. The cell is eventually going to run out of NAD+, a reactant of glycolysis. Because remember, in glycolysis, we are producing NADH. So this is an issue. NAD+ has now been turned into NADH through glycolysis. But in order to keep running glycolysis, we need more NAD+. We need more of that reactant. This is where fermentation is going to step in. Fermentation is going to be taking this NADH that we can no longer use and recycle it. Well, why can we no longer use it, you might be asking? Well, think about it. Remember the conditions that promote fermentation and anaerobic respiration. The most important one, that's probably the most obvious, is a lack of oxygen. If we do not have oxygen, all of those processes that occur in the mitochondria are no longer going to be working, including the pyruvate dehydrogenase complex, the Krebs cycle, and most importantly, the electron transport chain. The electron transport chain is the actual complex where NADH and FADH2, both of our electron carriers, are actually going to be used. All of these earlier processes, we're just producing electron carriers so that they can be used to produce ATP at the electron transport chain. So in this scenario where we're running fermentation and anaerobic respiration, the electron transport chain is off, and so we have no use for NADH anymore. Fermentation is going to be able to recycle it back into NAD+, a much needed reactant of glycolysis. That is going to allow us to keep running glycolysis, which will produce 2 ATP per glucose. Additionally, the Cori cycle, which like I mentioned earlier, we'll talk about after gluconeogenesis, is a cycle that will help us recycle lactic acid back into glucose. So we have a way to now recycle NADH, which we can't use, back into the much needed reactant NAD+, and we have a way to recycle pyruvate back into glucose. Again, this may seem like we are wasting NADH, but since we don't have any oxygen and we're not using the rest of aerobic respiration, we're not going to be using this NADH to form any ATP at the electron transport chain. So we're just going to recycle it so that we can allow glycolysis to continue. We need NAD+ as a reactant of glycolysis. This allows us to resupply it. When are we going to be switching to fermentation and anaerobic respiration instead of the pyruvate dehydrogenase complex? It's when we need ATP, but specifically we need ATP at a rate faster than aerobic respiration can provide, or just in flat out anaerobic conditions. If we don't have oxygen, we can't do aerobic respiration, we'll switch to the less efficient but faster fermentation and anaerobic respiration. The important enzyme that you'll need to know is lactate dehydrogenase, LDH for short. This is the enzyme that is going to be responsible for mammalian fermentation. Lactate dehydrogenase is the enzyme responsible for taking us from pyruvate and NADH into lactic acid and NAD+. You should be able to recognize that this is catalyzing a redox reaction. We are going to be reducing pyruvate into lactic acid so that we can oxidize NADH into NAD+. You don't need to know too much about the yeast side of fermentation, but I do want to remind you that yeast is eukaryotic like mammals, not prokaryotic or bacteria. For some reason, I always had an issue remembering that yeast was a eukaryote and not a prokaryote, so this is just a quick reminder that yeast is eukaryotic. You don't need to worry about this collection of enzymes. It's collectively called zymase that is used by yeast in fermentation. You only need to worry about LDH. So finally, just as a reminder, anaerobic respiration will be promoted by low energy, low ATP conditions. We need ATP. Additionally, it is going to be promoted by anaerobic conditions. We're lacking oxygen, or intense physiological activity where we need ATP at a faster rate than aerobic respiration can provide. Anaerobic respiration and fermentation will therefore be inhibited by high amounts of ATP. If we have a lot of ATP, then we don't need to be producing anymore. It will also be inhibited by aerobic conditions, oxygen is present, where our efficiency can be prioritized over speed, such as in low physiological activity. So now that we have two different pathways to produce ATP, one that is slower and more efficient but requires oxygen, and one that's faster and doesn't require oxygen but it's just not very efficient, this is going to bring up some advantages and disadvantages. The advantages of fermentation. The first is that it doesn't require oxygen and can operate when our oxygen stores are low. We are always going to be able to rely upon fermentation. Additionally, it produces ATP at a higher rate, meaning we can produce more ATP per second. It's not necessarily more efficient like we've been saying. In fact, it's less efficient. It's just producing ATP faster. The disadvantages of fermentation is that, of course, it is way less efficient than aerobic respiration. In fact, by a factor of 15 to 16. So we're going to be using glucose at a higher, less efficient rate. Additionally, lactic acid, or also known as lactate, is toxic to our cell. And so we're going to have to excrete it or recycle it after it has been produced. So think about whenever you're working out. I think most people intuitively know that lactic acid is one of the products of your muscles working really hard because your muscles are starting to do more fermentation. Your muscles will start to produce lactic acid, and that's why your muscles start to really hurt whenever you're exercising. This is post-production, Nate jumping in here just to clarify really quickly. Lactic acid is not necessarily the reason that your muscles hurt during workout, but it is still toxic to your cells, and it is something that our cells basically have to recycle. That's not really important for the purpose of the MCAT, but I did want to clarify just in case any of you were curious. This is simply just a byproduct of anaerobic respiration that our body's just going to have to figure out and how to deal with it. That is where the Cori cycle steps in. It's going to take that lactic acid back to the liver, turn it into glucose, and the liver will send that back to our muscle cells so they can continue doing anaerobic respiration. You also might have seen people use those massage guns, the really big black guns with a big ball on the end of it that vibrates really quickly back and forth, claiming that it removes lactic acid from their system so their muscles can heal like a day after they've worked out. That's actually false. Lactic acid has mostly cleared from your system within 15 minutes to an hour, usually. Inflammation, where your muscle cells are starting to repair themselves, is responsible for that soreness that you feel later in the day or the next day. Just one of those mother's tales that's actually false that I found interesting learning about. Another thing that I wanted to mention on this slide that's important for the MCAT, that the MCAT likes to ask about, is that lactic acid, of course, is an acid. It's properly named, meaning that whenever it is carried in the blood, whenever it has been produced and is going to be put into the blood, that is going to acidify your blood pH. And one of our body's reactions to a lower blood pH is to breathe faster. This is one of the many reasons that you start to hyperventilate whenever you're exercising. Your body is producing lactic acid. This is an acid that drops the pH of your blood. So through the bicarbonate buffer system, your body is going to breathe off more carbon dioxide to raise the pH of the blood back to normal conditions. If you're curious of why I included a picture of wine corks on the slide, that's simply just because we talked about ethanol on the last slide, how that's the product of fermentation of yeast. Ethanol is just one of the most common alcohols that we have in drinking alcohol. So that was just the picture that I decided to throw up here just to not make you go crazy only looking at text on a slide. So that's the pyruvate dehydrogenase complex as well as fermentation. Just as a reminder of what's going on in the both of them. In the pyruvate dehydrogenase complex on the left, this is going to feed into the rest of aerobic respiration. Specifically within the pyruvate dehydrogenase complex, we are using that enzyme pyruvate dehydrogenase. That's going to help us produce 1 carbon dioxide, 1 acetyl CoA, and 1 NADH per pyruvate, just as a reminder of the summary of the PDC. It is going to occur in aerobic conditions, since this is in the mitochondria. It is also going to occur in the postprandial state, where we have high amounts of glucose in the blood. We're doing a lot of glycolysis to produce pyruvate, and we need ATP. We have low energy stores. Our ATP need also is not desperate, and our oxygen levels are normal. This is going to allow us to run the PDC and the rest of aerobic respiration. It is also going to help us through the rest of aerobic respiration produce that really efficient 30 to 32 ATP per glucose. Fermentation, on the other hand, in addition to the Cori cycle, which we'll talk about later, is going to occur in very similar conditions when we have a need for ATP and a ton of glucose to provide that ATP. But we either do not have oxygen or our ATP need is faster than what aerobic respiration can provide. This is going to occur in the cytosol, the anaerobic conditions in the cytosol. And it is going to produce only 2 ATP per glucose. The most important enzyme you need to know for fermentation is LDH, lactic acid or lactate dehydrogenase, which is going to take pyruvate and reduce it into lactic acid, which is going to allow us to take NADH, which we no longer need in this condition, and oxidize it into NAD+. That's going to allow NAD+ to go back into the reactant pool for glycolysis so that we can keep running glycolysis at that very fast rate.